So, didn't want to leave you guys hanging for your Biology 2 labs. I already gave you the pre-lab for Plant Lab number 1, so this is going to be for Plant Lab number 2. Again, all the other material for lecture is already on Canvas. All the PowerPoints are there. Everything is sitting there waiting on you. So just to reiterate, just so in case you need to read the announcements as I post them and you need to follow them, just to make sure that the chaos is limited, especially on my end, to try to keep everybody's work together and you get proper credit. And also please abide by the timelines because otherwise it becomes a mess. Alright, so for your second lab, it's going to be for the higher plants, meaning those which are seed-bearing plants, meaning cone-bearing plants, and then the flowering plants. Alright, so for those, I'm going to make my really bad drawings for you. Alright, so the first one, probably better if I do this. First one is going to be the what's called the Pinus 5 needle. Think of a pine needle, the length of it, cut it straight down. It kind of looks like pieces of a pizza pie. It's very symmetric. Also along with that, if you ever play Trivial Pursuit, it looks like one of the game pieces. The one I put for you on Canvas is only one of the slices, but the actual pine needle looks like, oops. It would look like a bunch of wedges coming to a common point, kind of like a pizza pie or, the Trivial Pursuit game piece. The one I have for you is one of these blown up. So if I blow this one up, it's going to look like this. What you'll see toward the outside, that's going to be the epidermis, obviously. That's going to be the outside layer. You're going to see a large circle maybe in there. That's called a resin duct. That's where the pine needle will give off the compound resin, which basically it's so that other creatures won't eat them. It basically tastes foul and it's a toxin. Coming down in then, you'll see a circle right about here. And on that circle, that's going to be the vascular cylinder. Toward the middle, you'll see large big cells right here. That's going to be xylem to move water. Out here, you'll see smaller cells out toward the outside. That's going to be phloem to move food. All around the whole area, what you'll see are a bunch of cells like bubbles all over around all of it. That's the chlorenchyma. Those are the chlorophyll-bearing cells that will do photosynthesis, hence why the pine needle looks green. All right. Next thing in the the pine tree that you have to draw, I'm trying to do this all from memory, so apologize, is the pinus stem. The stem, basically think of like looking at a tree when it's cut down, the tree rings. That's more or less what we're looking at. So over here then, you'd have a series of like rings coming out like this. And there's going to be these huge dark cells going all over it. All of that is xylem, okay? So the oldest part of the stem obviously is in the middle. That's where you would call it the quote heartwood if it were old enough, where most of the water's been drained out. It's the hardest part of the tree at that point. As you get further and further out, these cells haven't had the chance to compress yet and to get all the water drained out yet, so they're often called sapwood. I mentioned this in class. It's kind of like whenever you when you go to a Lowe's and you buy lumber and you pick up a piece of like a 2x4, it's pine, and if it feels moist or damp, well, that's from the cheaper part of the tree, the sapwood, and therefore it tends to bow when you build with it. The expensive stuff is the heartwood stuff. All right. Then what you'll see as you go further out, you're going to have a bark line out here. Okay. That bark line you'll see is one of the options listed as part of what's called the phyloderm. That's going to be old cork cells and old phloem. So basically kind of like your skin. You don't see rings of phloem because it sloughs off just like your skin would slough off of you. The most important area is going to be this area right here underneath that bark line. Now understand, in the bark line, yeah, you'll see large holes. Those will be resin ducts like they were in the leaf, but it's really unimportant. This area right here is going to be really important. Why? Because there's going to be, you're going to see like a green layer right above on the outside right there, and then you're going to see the red cells on the other side. That's an important ring because that's the ring where you have live xylem toward the inside of it, the green cells on the outside of the live phloem. They're the only ones that are living. In between them is a layer of tissue called the vascular cambium. The vascular cambium is their stem cells for vascular tissue. And as those cells divide, you make new xylem toward the middle, new phloem toward the outside. So that part is an important part. If you were to remove that whole vascular cambium around the trunk of a tree, that's called girdling a tree, and you essentially slit its throat. It can't survive. So the food coming down dumps out onto the roots. The water that comes up out of the roots comes up and then dumps back down the roots. If you effectively killed the tree. Now, so that's the stem. Next thing, if I remember right, then we have what is called the megastrobilus and the microstrobilus. Strobilus in Latin is essentially saying cone-shaped. So the microstrobilus is the male, therefore he's going to make pollen. The female is the megastrobilus, and therefore she's going to make the ovules. Now, whenever you see the words microsporangia, microsporangia means essentially the stem cells, which would lead to the male counterpart, anther. So, sorry, the pollen. Whenever you hear megasporangia, megasporangia means that that's going to be the stem tissue leading to her ovule. All right. So what I'm going to do is I'm going to make one pine cone. Okay. On the left is going to be the boy. On the right is going to be the girl. All right. So again, we're just going to curl up like that. Now understand on your lab it's two separate drawings. So down the middle, we'll put the male over here, the female over there. Each level is called a scale, okay? Now, in the male, when you go all the way to the bottom of the scale, there'll be these large pouches right here near the central shaft, and it'll have little grains in it. Well, these little pouches right here, that's the microsporangia. The little grains are the pollen. Simple enough. On the female, same thing. When you come in, there'll be these large balls on the inside. Now, what you'll see is that there'll be kind of like a smaller ball inside of this. The large ball is the mother megaspore, meaning the tissue that leads to it. The little dot is the ovule, the egg. What you'll see coming down to it is an opening that comes down to it. The opening is called a micropyle. The micropyle is when the pollen would attach to the female. That's where the pollen tube would travel down till eventually fertilize the female. Okay. Last but not least, for the pine, if I remember right, it's called the pine embryo. So think of like a seed shape, okay? So you're going to have a seed coat like this on the outside. In general, it's just called integument. No big deal. Now on the inside, whenever I teach this, when I would be with you, you got to think of the embryo has a general body form. Most plants have the same general embryonic body form. I always call it like a satanic bunny rabbit. It's what it looks like. It looks like a rabbit with horns, all right? But again, the horns can be shaped differently, but it's the general pattern. All right. So what you're going to see is this little structure on the inside that looks like a little satanic bunny rabbit. Okay. Now, this part down here, that is the embryonic root. That's called the radicle. Whenever the seed absorbs enough water, it will swell, and it's the first thing that's going to come out in order for the seed to germinate. This little bump in the middle, the head of the satanic bunny rabbit, is called the hypocaudal. The hypocaudal is the embryonic stem. These two bunny ears, those are the embryonic leaves. Those are called cotyledons. Understand when the seed is underground, it cannot do photosynthesis. It's underground. So therefore, it has to have a means to get energy, and therefore the cotyledons are essentially acting like the yolk sac for the embryonic plant until it can actually make its own leaves and photosynthesize and then grow on from there on out. I'm trying to think. I think that covers everything for the pine trees. Okay. So what we're now going to go into next is the second half of the lab, which is going between monocot and dicot. Okay. So in your PowerPoint, what I gave you is a one-slide PowerPoint that has comparing monocot and dicot. I cannot stress this enough. Know it. It's one of a key concept in biology, too, is knowing between mono and dicot. Real simple when you think this in your brain. Monocot, think corn, think a blade of grass, think a lily. Usually that usually helps you out if you can picture those. And when you think of a dicot, think pretty much any goddamn thing else you can mention. Think of like the weeds in your backyard, daisies, think roses, think, the list goes on and on and on. Most things are going to be dicots. Okay, so both of these, understand, are going to have seeds. Both of them are going to bear flowers. One form, well, most of them are going to bear flowers. All right, so first thing I have you look at is the difference between a monocot root and a dicot root. So I'm going to put monocot here, dicot here. All right. Now, the picture I drew for you in class before we had this whole fiasco, if you remember, is the same general plan. You have a ring on the outside. That's going to be the epidermis. Then you're going to have this space till you get all the way in the middle. That's going to be called the cortex. It's a storage area for water, food, whatever. Now, the most important part is going to be that little circle in the middle. That's called the vascular cylinder or the stele. That's where the xylem and phloem are located. How they're arranged is going to determine whether or not it's a monocot or a dicot. Okay? So, this is your epidermis. That's your endodermis on each side. Now I'm going to take this and I'm going to blow it up so you can look at it. So that's the stele blown up. All right? What you would see is going around in a circle, large circles going around in a circle. Usually they're stained dark red. And then what you'd see, is kind of like there's large red cells in them. That's the xylem. Okay? Now, in between where these circles are, you're going to see little groups of cells in between. But they're more toward the outside. Those would be phloem. Okay, so it's always xylem more toward the middle, phloem toward the outside. So it's kind of staggered like a zigzag in a circle. One circle. Really important too, there is no layer of tissue between them called vascular cambium, which means this root cannot show secondary growth. Secondary growth means growth in girth. Think of like when you see a baby carrot versus an adult carrot, it gets fatter over time. That's a dicot. Think of like the root if you ever pulled a blade of grass out of the ground. These little thin fibrous roots, they don't get thicker over time because they don't have the tissue to be able to do that. Now, over here on dicot, if I take this, actually I can do it on here. Don't need to blow it up. In the middle, the xylem will give you like a star pattern. Because my artistic qualities are very limited, keep in mind I'm going to make a very simple star, a 4-pointed star. Now I can have 6 points, 8 points, 12 points. 4 is my limit. 4 is simple. In the middle, what you'll then see are large cells in the middle. That's xylem. Tucked in between the arms of the star, you would see cells here, 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 and here. That's phloem, toward the outside. Important. Between the phloem here and the xylem here, there's a layer of tissue surrounding the star. That's the vascular cambium. That's the stem cell, so the root can get fatter over time. Okay? So that's the root. Let's then move up, go to the stem. It's getting messy. All right, so the stem. Again, I'll put monocot here, dicot here. All right. For a monocot, real simple. Again, think like an asparagus, like a corn, like I said before. You're going to have an epidermis on the outside. You're going to have the cortex surrounding on the inside for storage. Most importantly, the vascular bundles, the xylem and the phloem, will be these little scattered little beans all around on the inside. So they're scattered. No particular geometry to them. If I took, say, this one and I blew it up like that, notice this would be pointing toward the outside, that would be pointing to the inside. What would you see? You would see this little circle right here, and that's your phloem. All around it, you would have these big cells, and that would be your xylem. So it's always xylem toward the middle, phloem toward the outside. Now, over here for a dicot. For a dicot stem, that's kind of like the same stem that you think about whenever I drew the pine tree. The rings coming out. The one that I gave to you is for ranunculus, which is for a dandelion. They don't bear wood. They don't have bark, so they're not going to have that barky phyloderm on the outside, so they won't have the discerned rings. So for the dicot in this one, what you'd see is, again, you'd have the center of it, often called the pith. You'd have the epidermis around the outside. The most important thing I need you to see is right underneath the epidermis is going to be this ring. That's going to be the vascular cambium. What you will then see spaced all the way or evenly around is going to be a bundle on the outside, bundle on the inside, bundle on the outside, bundle going all the way around. On the outside, what do you think it is? Phloem. On the inside, what do you think it is? Xylem. Okay, I know I'm beating a dead horse here, but I'm trying to pound this into your head as best I can. All right, that's the stems. Moving on. Eraser. We now go to the leaf. Now, monocot. Again, picture with your brain, you went out to your front yard, you pulled out a blade of grass and looked at it. Notice when you look at it, the veins run parallel. Think of like a corn husk. The veins run parallel. So what you would see for a monocot leaf, basically, I'm only going to draw half of it. You'd have this middle central part, and it would kind of taper as it went to the edge. All right? Now, it would be the same thing on this side, but, just it's be symmetric. What you would see going out onto this blade, you would see a series of circles going out all the way out. Those are the veins. That's the vascular cylinder, the tissues. So what would you see? Large cells in the middle, xylem, small cells toward the outside of those circles, phloem. Surrounding it, what you would see are scattered cells surrounding these circles. That's the chlorenchyma, the chlorophyll cells. That's why the leaf is green. On the outside, then, you'd have the epidermis, obviously. Now, the thing about this is on the epidermis, if I took the epidermis and zoomed it in a little bit, what you would see, it would look like a bunch of little bricks going one after another. Those are regular cells. Every so often, you're going to see two cells kind of get small and pinch off, and then it goes back to a brick road again. Those two little cells that are pinching off are the guard cells. The hole in the middle would be the stomate, the stomata, the leaf pore for gas exchange. So therefore, these two guard cells, when they inflate, they close the stomata. So when they inflate with water pressure, they pop open the stomata. When they deflate, they close the stomata. All right, that's for the monocot. For the dicot, this one I drew for you when we went over the basics of photosynthesis. And I said when you look at that leaf, it kind of looks like layers of a lasagna or like layers of a cake. So you're going to have this outer upper layer. That's going to be your epidermis. Let's assume the sun is coming from this direction. Okay, that would be the side facing the sun. Well, then I'd have the same thing down here. That would be in the shade. Okay, that'd be the underside of the leaf. Both of those are the epidermis. Now, coming off of the epidermis, you'd have these long, skinny cells that kind of look like stalactites coming off. And they're all uninucleated. That's going to be the palisade mesophyll. That's the chlorophyll cells. That's why the upper side of the leaf looks green. Take the leaf, flip it upside down, doesn't look green anymore. Why? Because you're going to have this loose aggregation of cells on the underside. It kind of looks like a dried out kitchen sponge, and that's why it's called the spongy mesophyll. Meso means middle, fill means layers. Nothing really crazy. Now again, down here on the bottom row, every so often, kind of like over here in the monocot, these bricks will pinch off to make these little cells, but then go back to a brick road again. Again, those are the guard cells. The stomate is the hole, is the space in the middle. That should do it for the plant lab. Except for one more thing before I forget, you need to know the anatomy of a flower. So, flowers are going to occur in what are called four whorls. Meaning if you look down at a flower, you would see four circles, kind of like a dartboard going toward the middle. All right, so again, picture in your mind a flower. You pick the flower, take the flower, flip it upside down. You see that green ball on the underside. That's the first whorl, okay? What you got to remember is when that thing was a bud, that was sealed. And therefore, when the water pressure builds up, it pops open and then the petals pop open, all right? So you got to think of it before it was a flower. So here we have the stem coming up, okay? The stem will then make that ball at the bottom. That's called the receptacle because you're receiving the flower, okay? Now, on the underside of this, if you think of a flower, you see these little tiny green leaflets that are on the underside. Well, that was the remnant of what used to be the bud before it popped open, okay? That's going to be the outermost whorl. If we go all the way, well, each one of them is going to be called a sepal. If we go all the way around in one circle, that's called a calyx. Okay, so one of them is called a sepal, all of them is called the calyx. Now we go to the next layer in. That's what you're used to when you think of a flower. The petals go, oh, it's so pretty. That's that one, okay? So those are your petals. like that. All right? So that's the second whorl coming in. Now understand there are a specific color, a specific shape, a specific pattern, a specific fragrance, because they're trying to attract a specific pollinator. They're talking to them chemically and visually to say, here, come here, take my pollen, go fertilize, and try to basically manipulate life to do your reproduction for you. Okay? If these petals are in groups of threes, meaning in multiples of three, that's a signal of a monocot. If they're in multiples of 4 and 5, that's a dicot. All right? Now, all the way in the middle, then, we're now going to come to the male part. The male part is called the androecium, meaning the boy part, andros. What you'll see is these little things coming out with a little ball on the end. If I take that and blow it out, there's the boy. The whole boy is called an anther. Sorry, stamen, my bad. Again, I'm trying to go too fast. So the whole thing is called a stamen. The little ball up here that makes the pollen, that's called the anther. This little thing that makes it stick out is called the filament. The two together make up the stamen. The stamen is the male. Smack dab in the middle, then, you have this vase-like structure that comes all the way down, like a vase, and that's going to be the female, like that, okay? If I take her out, she looks like this, okay? She is now going to be the gynoecium. Again, he's the androecium, she's the gynoecium. The whole thing is called a pistil, P-I-S-T-I-L. This top part up here is sticky. It's called the stigma. This part right here is called the style. Down here, then, you would have your ovules, your eggs. We'll understand why that part is sticky. Pollen is going to be done either through the air or through a pollinator. The pollen attaches here, and then the pollen has to travel down the style to fertilize the eggs. That's why it's sticky. All right, that's the flower anatomy, and now I think we're officially done with that pre-lab. Thank you very much.